Today we have the pleasure of welcoming Dr. Kai from Texas A&M University. Actually, his department is integrative veterinary integrative biosciences, but he has a lot of affiliation, including ECE department, genetics, statistical, bioinformatics, exactly. So far in the course, I was explaining to Dr. Kai, you learned about proteins, sequencing, computational biology, AI in healthcare. So this is really perfect lecture to finish that pillar of the course. His research sits right at the intersection of all these areas. His work combines human genetics, data science, and advanced computational methods to understand how cells behave and why individuals differ in their sensitivity to diseases. So it's really interesting because it's going to talk really to the single cell level. I mean, isn't that incredibly fascinating? And this is really, this type of analysis is central to every modern precision medicine. So Dr. Kai has co-authored over 200 publications, 2 chapters book, and his work has been cited thousands of times, reflecting really the broad impact across genetics, computational biology, and machine learning. So without further ado, please welcome in here, Dr. Kai.
Thank you for the introduction. Okay, good afternoon. So I'm from vet school, and my background is general medicine, biology, and then get into the computational biology side when I become faculty here. I also affiliate member from ECEN, and my best students all from ECEN, and they are interdisciplinary research. So today we are going to talk about single cell and gene expression analysis. So this technology has been rapidly evolved over the last decades, and especially, and already 10 years. And I just cannot imagine that when I started getting the data ourselves in 2017, 2018, and very quickly, it's already almost 10 years. But at the very beginning, and the biologists only can, in their lab, to do 1 or 2 cells and 10 cells, 100 cells each time. But in the year 2017, and there's a rapid increase. This is by scale, log scale. In 2018, the journals believe it's a good time to claim single cell revolution is starting. So this is a breakthrough of the year 2018 by Science Journal. I believe this is a number of publications of the. So one of the day in 2017, I threw out all the paper I previously print out before the single cell gene expression data become available, because they all so-called bulk RNA-seq and gene expression sometimes by microarray. And I will show you why they're not informative enough. So all the conclusions, evidence, analysis approach may be wrong and misleading. Okay, so I recycled all of these papers. If you know this transformer language model, this structure framework, so machine learning actually also get this revolutionary technology. Transformer was the framework was introduced about 2017. So I believe there's an intersection of these 2 major technology revolutions at that year. Again, this is an illustration for the technology development. We completely driven by this revolution. And this is a 2017, the introduction of this small machine by 10x Genomics. It's a company, California-based. So they introduced this controller into the market. What this machine can do is to load your samples, 8 at the same time. And then they create this so-called droplets, okay? if you can see, there's an individual cell and this black bubble here, black ball here, and will be encaptured into a oil droplet. So in that way, individual cells will be separated. Each bubble capture one cell on average. So this is the channel, and this is a captured droplet, and you can see this is this two droplet contain individual cell, and this is empty. So at the end, we will break down the membrane of cells, release all the barcoded individual mRNA molecules. They are indicator of the gene expression activities. Because of this isolation and the barcoding system used for each droplet differently, so there are unique barcode for each individual cells and all the mRNA molecules in there. So this is a zoom in. You can see this oil droplet look like this, and then you have a cell has been captured. And sometimes you capture two cells, and then you double your expression level. You will clean up later using bioinformatics. You know there are multiple cells captured. But if you dilute enough, you should, on average, you always capture majority of a single cell. They're empty droplet, and because you don't detect anything, and you will filter them out later. Anyway, so in the center of this is a gel bead covered by a barcoding system. And so everything released from this particular individual cell will be labeled with this barcoding, this DNA fragments. You can consider this as a contamination. So you contain all the cell components, all the material released from this cell with some contamination, so barcoding. And different type of contamination tell you, okay, this cell expression in this way, and that cell expression in that way. So that's the barcoding system. So this is not only solution, there are also split two combinatory barcoding protocol, and another company, as an emerging company also called, come up to the market. But for now, the market will be, say, 90% is dominated by this droplet-based cell partitioning and the barcoding system. So we are going to focus on this. For the downstream analysis, this other technology will have a very similar framework for the analysis, because bioinformatics today, we don't know their hardware, the technology, how this information was collected. So this is a bulk RNA-seq. That's a paper that generated from this technology. I throw away, trash them, okay? So when you have a bulk RNA-seq, you smash your samples and extract RNAs. Then you get this particular sample's expression of the gene. So for human gene, we consider there are 10,000 genes expressed and maybe more. And but that's only one sample, and it's a mixture of different cell type. So it's give you an average, but not enough resolution. The foundation unit of this fundamental unit of life is individual cells. So that's why this 10x genomics introduced this cell barcoding system, allow us to have this dissociated tissues, to separate them, use enzyme to digest, to make this cell suspension, and each cell separated from another. If it's a blood cell, it's very easy. You don't have to do even separation. You just load the cells into this barcoding system and use this droplet to capture all the individual cells. At the end, after sequencing and after this bioinformatics processing, you will get a matrix, okay? So this matrix contains thousands of cells, expression of thousands of genes, okay? It was bulk RNA-seq, one sample, just one expression, but now you have all the cells expression of up to 10,000 of genes expression you can detect. So from a data science perspective, we are very happy to have the matrix rather than a vector to play with. Okay, so this is high dimensional data, and each sample, say each cell, is defined by their gene expression profile of thousands of genes with thousands of dimensions you can use. So that's I put that trashed paper again, okay? So I really don't read them anymore. So I convert this heat map into something more visually appealing. So this is I call it a bird eyes view of gene expression landscape. Remember, this is only a small corner of huge matrix. I said 10,000 by 10,000, 10,000 cells have the gene expression data in 10,000 genes. So this is a very small corner of that huge matrix, but you can start to realize genes have different levels of expression. For example, this gene expression level is higher than those genes here. If you look at by cell, and each cell is defined by those peaks and high or low, a lot of zeros, but for each individual cell, they tend to have a very different profiles and to distinguish them from each other. So our overall goal is to work on this matrix to understand the landscape. For example, if you have a feature has been summarized in these bars, so now it's not just single cells expression, single genes. It can be synthesized for several genes together, a kind of profile quantity, and you can define, okay, this is a cell in the diseased stage. Well, you also, from the same samples, also have a healthy cell state. In ideal situation, we want to introduce perturbation, maybe a drug, or maybe small molecule lead to push the cell states to healthy states from a diseased part. So this is some kind of overall landscape view. Remember, we can do this very easily to load two samples if you have healthy control and also against the disease. Or in this example, you have two samples. One is from a wild type before the mutation, and this is a introduced knockout samples. You knock out particular genes, and you collect the animal samples from this knockout animal. Then you have two matrices, okay? So this is, we are even happier with the two matrices to play as a data scientist, as a computer programmers. So this is like illustration of two samples, blue cells against red cell. And you can start to appreciate this landscape change, their overall downstream upregulation for the orange cells, and they're more dispersed or gene diversity or gene expression variability among blue cells. So not only mean, but also the variability change is the more important sometimes in single cell data analysis. So from 2017, we start to build single cell data analysis pipelines. For example, we have a single cell RNA-seq data input, and then their dimensionality reduction technique. I believe a lot of you knows t-SNE, UMAP. They are not exclusively used in single cell world, but also in the other data mining and the data visualization technique for high dimensional data. But we commonly use those and to build those visualization, and we can have a FATE, which is a new technology, and we can have trajectory. So you follow the progress of the cell from one stage move to another, and you build this trajectory. Believe there are some of the time series image signals along this trajectory. It's called a pseudo time analysis because they are not real. They just align the same cell, same sample of cells from a single time point collection, but you try to reconstruct this development stage. You can build a gene regulatory network using single cell data, and then you can put a network along with pseudo time. That's because you have enough cells, you can do whatever you want. You're not limited by the number of samples. And you can have a dynamic modeling to simulate this change of the network from one time point to another. So a lot of things we can do. we also developed this toolbox, have an interface. That's essential because we wanted molecular biology labs, the people have no coding experience can use this very sophisticated operations, manipulations of data points. So we provide this interface, single cell gene expression analysis tool, allow them to utilize this MATLAB-based interface to convert the matrix into something make more sense to them. So this is a dimensionality reduction use 3D t-SNE plot to generate the data. There are samples from 2 different batches, and you see the different colors, blue and orange. And because it's 3D, so biologists can drag and visualize how whether there are small cluster of cells, whether this is a rare cell type have never been discovered in biology field, so they're all possible. The most important step is to do the clustering and then to do the annotation, see what cell type in your sample. If you have a cell cultured cell, and then all the cultured cells are from a similar clones and they are same cell type, but something like this, they are indicators that this is from a primary tissue, from the animal itself. So you have a mixed fibroblast, you have all the immune cells, macrophages, neutrophils. So, but once you put this cell type and different batch IDs side by side, they have this multi-dimensional view to help you to understand the change of composition of cells, so mix different types of composition of cell type between P1 and the P2 samples. So there are many different ways once you have these different kinds of the separation and visualization of data. Okay, so another we can do is to extract particular cells. For example, you are interested in only neurons, and then you can have a neuron-specific analysis downstream because you can get rid of all the oligo cells and only focus on the neurons. So single cell data allow you to do cell type specific analysis, which was not possible when you have a bulk RNA-seq. You have a microarray data from a mixed of cells. So that's maybe 80% of the neurons, 20% of oligos, dendrocytes, and sometimes you sample it differently. They have 20% of neuron, 80% of other cell types. So that's not very informative, but now you have real focus on which cell type. If you build anything, say gene regulatory network from neurons, that's between neurons, and you use the population size to approximate the individual cells regulation pathways inside individual cells. Okay, overall, you can use single cell technology to do cell type annotation and to identify rare cell types. You can build differentiation pathway or trajectory analysis. You can have gene expression program analysis. You can have also their spatial transcriptome data now allow you to keep the original tissue structure, but also sample at a single cell level. In that case, you have information about which cell type is located next to each other and which cell type are not close to each other. And you have some power to analyze this cell-cell communications. Even without this physical contact, we have a way to do the cell-cell communication analysis. I show you later how to do it. Okay, the overall challenge is you have to try to get the information which reached but head into this static image. You want to reconstruct something dynamic of the cell gene expression program, want to reconstruct the trajectory. by looking at the data from just one single shot of the time. I also illustrated with this picture, and in order to take this picture, you have to take a series of pictures and put them together later. The single cell data gave you this information. You just have to, the contents of the cells in different stage. You just have to understand analytically what they are and how they organize in terms of time, if you are interested in time. So this is an illustration of the spatial transcriptome I just described. So you have this sample, it's a cross-section cut for the a slide of tissue, and then you have information of the individual cell level resolution expression and in that. This is to illustrate further how you reconstruct this pseudo time to give you information of the dynamics. So if you illustrate all the cells, just use t-SNE without construct pseudo time, you don't have that information. But if you put the population of picture of the horse together, and they all at a different this gate position, and you probably can reconstruct this video in series of this picture. So the same idea. So you try to look at one single cell population at one single time that reconstruct the trajectories. It's a revolution for every subfield in the biomedical research. And if you look at the papers, single cell technology or single cell RNA-seq search, you get all kinds of papers published overwhelmingly. And also it's getting evolved further, and not only the expression level. So what we just discussed is so-called single cell RNA-seq. That's only gene expression, but this is a major one, of course. But you can also have other informations about open chromatin structure, which this is tell you which part of this chromatin is open or ready for regulatory element to bind to regulate expression. And you can also have a methylation information. This is called epigenetics. You have a DNA. If you have a particular point mutation, this will be able to give to you for that particular cell. If you trace the cancer colony and development, this may be very useful to catch the driver mutations. Anyways, sometimes the same cell, you will get both expression and attack-seq at the same time. This is called a multimodal data analysis. In that case, you will have not only one matrix from that particular sample, but for the same group of cell, you have two matrices. One is RNA-seq, another attack-seq. So this is a very challenging analysis task. So this is some products from our lab. So the interface, very important to communicate with collaborators. We build pipelines to do analysis. We also introduced quantum computing to capture some of those high dimensional information simultaneously. So I will go through this pipeline first, and we'll touch base on some quantum machine learning field. Okay, so my lab now is working on so-called single cell systems biology. When you talk about the systems biology, systems, you need to build networks to understand the different components relationship. So I'm going to go through several tools we build using machine learning approach. So the first tool, this will be used to construct single cell gene regulatory network and then compare them. So construct one network from one sample, construct another, and compare, and let's see what's the difference in their network. This is called a single cell 24-net. As you know, you can have this matrix, and that's what matrix looks like. That's all just count, how many copies of that gene expressed in each individual cells. So you can have a Excel spreadsheet to hold this data. What we would like to do is to look at the pairwise of the genes and then to look at their relationship. Each point here is a cell, and these cells express both gene high, and then these cells express both gene low. So this gives you the correlation. So you can use correlation coefficient to define the relationship between this is positive correlated. However, if you look at the population of a cell and look at their scatter plot for these two genes, you see a nonlinear relationship. But if you further delineate this by using the third gene, so those cells have a lighter color of this PRDM1, indicate this gene, those cells have a high expression of this third gene. In that case, these two genes are positively correlated. Well, with the refined subpopulation, this third gene, PRDM1 expression low, then these two genes look like negative correlated. So this introduced the some co-independent between three genes, but if you consider the gene expression network contains thousands of genes, but very complicated. But it gave you some hope that with enough cells, you will be able to look at pairwise gene expression correlation, and then considering other parts of the network, maybe you can figure out more complicated or more realistic regulation relationship between multiple genes. So this is the example, and we can really use the one single samples, because single samples contains thousands of cells, to reconstruct so-called NF-kappa B, is a very important immunocell regulatory network. This picture has been built by classical experiments using animals, using cell cultures, but we can almost use a single cell data to reconstruct the whole regulatory network. So that's a demonstration of this all pairwise correlation. They all can be predicted correctly with two exceptions. So our goal is to use single cell data, try to construct the gene regulatory network for two samples. If I construct one sample from condition 1 cells compared to regulatory network from condition 2, and what we want to find are those genes, for example, gene number 2, their relationship was strong with these three genes, but become weaker with these two genes when the condition changed. So we want to identify this so-called differentially regulated genes by compare the networks. So for this pipeline, it's called 24-night, and this is overall pipeline. I will very quickly go through A, B, C, D, E, 5 steps. So that's the samples. You have expression matrix from one sample, and then there's a second. That's input. We will have first step, we do some cell subsampling to make subsampled copy, bootstrapping, and then use PC regression, principal component regression, to build gene regulatory network for each subsampled this data, and then use tensor decomposition to do the decomposition and then reconstruct a denoised version of the network. Then use this manifold alignment to compare to find the genes. So this is the first step to do the cell subsampling. We can define how many subsample you want to recreate. The more you will have more denoised effects, but you increase the computational burden. After then we have PC regression method to construct a series of the networks from each subsampled cells and put them together. And this is a tensor decomposition. Oh, by the way, this is a accuracy for the PC regression compared to other methods. So next is to do this tensor decomposition to learn the major components and then reconstruct by getting rid of this trivial components. So this is a matrix decomposition, and this is an illustration for the tensor decomposition. There's a use for tensor decomposition to do this feature detection or denoise technique. I believe you may know this better than me. This is examples. So how many components you will keep? If you keep 10 components, your network after denoise looks like this. But if you only keep top 3 components, looks less noisy than once you want to keep more components in the reconstructing procedure. Anyway, so this is one sample and another sample. This is constructed networks, and this is denoised version. So we are going to compare this to denoised version of the network through nonlinear manifold alignment. So just treat this as the data itself, try to align them into a manifold space. And then we try to find this gene which changed their, cannot be aligned the best. So if you cannot align, that means your position in the network differed from your position from another sample. So those genes will be prioritized, put on the top of our list. Those genes are the so-called differentially regulated gene. That's a difference between 2 networks. So that's for biology students. So what is manifold? So what is after we detect those differentially regulated genes. So those genes will be collected. Say I want to keep top 20 of this highly dysregulated genes. I will have this gene list and to do downstream functional analysis. In bioinformatics, you have many different ways to do gene set-based functional analysis. Use the GC, enricher, they all pathway, so this overlapping test, basically. So you can detect, okay, what's the difference between these 2 networks? What are genes response for those difference? So applications, we apply our method to a data for other people have already published. What they did is to have a neuron, individual data from the controlled mouse against the neuron from the mouse has been challenged with morphine. So you expect if you have morphine injected to the mouse, the neuron will change its expression compared to those you didn't inject that. But what they found is these 2 groups of neurons before and after injection of morphine, there's no major changes. If you look at those differential expression, they don't have enough time to upregulate gene expression at all. You don't see this, so the author of this paper actually gave up. They say, okay, there's no neuron difference in their expression. They try to find some other evidence to publish this paper. You can take a look. But our method, we can compare the mock treated with morphine challenged neuron gene regulatory network, and we can find those places that regulation changed the most. Then we put these top genes through the functional analysis. The keywords come back for the function of those genes getting involved are morphine addiction, opium signaling, gamma alpha beta receptor activity. So they all related to the regulation of those morphine response. And it's really good evidence. Our method, which really powerful to detect this subtle difference in the regulation change rather than the average expression level change. The people also we can use in another examples to find the genes that are involved in different biological pathways, including Alzheimer's disease, other development stage of the cells. Okay, so the next tool we build is for so-called virtual knockout. it's based on the gene regulatory network. Then we simulate if I have one gene is knocked out, which disable that gene's function, what will happen for the network? You know, in the gene functional study, people use knockout animal all the time to knock out that gene and see what other genes change their expression. But this is really tedious biological process. You need to get the knockout animal strains and grow them and then get the tissue, do the expression analysis again to compare the wild type. But what we here, we want to use a simulation to do that. So we use data from wild type. So you see this pipeline was a network comparison pipeline. The only difference is we are not going to use the second condition, no second sample, only use one sample. What we did is make a copy. So these two are exactly the same. Then you can go through this pipeline. You will get some random genes out because they're identical inputs. So anything out should be a noise or the stochastic process in the middle. But what we did is to artificially remove a gene by changing the gene expression level to 0 for that gene across all the cells. In that way, we create a simulated knockout samples. And then we compare these two samples using exactly the same pipeline we built for the network comparison. That will give you the top genes. Those genes, the first gene will be the gene you knocked out, okay, all the time. But the second gene and the third gene and the next will give you a spectrum of this gene, which under influence of this knockout effects. Later, we also make a quick way to knock out that gene directly from the network itself, rather than through the raw data. So that's another way to accelerate this. But using the same idea, we can identify genes which, after you knock out, what other genes will change its expression or response, regulatory relationship differently before knockout that gene. So we use one example to knock out the TRIM2 genes from a mouse neuron microglia. Okay, so this is data we collect from other people published data and then predict after knockout the TRIM2 genes, then the gene get involved in Alzheimer's disease, oxidative phosphorylation, lysosome, and cholesterol metabolism. Those genes should change their expression or change their relationship with the TRIM2 genes knockout. Then we compare the animal study, the original paper published. So they create this knockout TRIM2 knockout mouse model. And then they measure the gene expression change compared to the wild type before knockout. What they get is exactly the same pathways of the function of those genes. So that information tells us they shouldn't do the knockout animal. They should do the virtual knockout first to predict the consequence of the experiment. If we can do this, we probably save a lot of life of the animal's life. But anyway, so this is the idea to replace knockout animal use by introduce virtual knockout framework to get this similar pathway of biological understanding, at least at a single, at a pathway level or gene site level, function group level, what you can have. So these two tools we just described, the compare network and virtual knockout, rely on to construct a good network. And just like if you have a good network and very accurate, and you perturb this network, you know which place you get. So people are start to using the virtual knockout tools. And this is a Nature Biotechnology paper. They identified a small molecule to have this clinical value for the fibrosis. And then they use KNK virtual knockout to simulate if they knock out this target gene and what will happen, and to validate their discoveries. this is another paper, and to apply to a different system. This is the stem muscle-like cells. So different systems. So there's a smooth muscle cells and different genes, and they found it is very useful to use virtual knockout to simulate to be informative enough. There are other papers built upon this knockout ideas to find the differential trajectory for other studies. We also have other tools. For example, I'm not going to any details for those tools. I just very quickly go through. So virtual knockout, a similar idea, and you can help you to fix the problem, so-called survivorship bias. And in biology field, they always have to have a wild type against knockout sample. You have to have this sample. You have it as a live cell to compare with. But if you have a mutation, kill the samples, but you force the cell to growth, all your collected samples are survivors. They are not a real function. They are not a good representative to reflect the function of that particular mutation or gene you knocked out. They are information gave to you, basically. So if you use virtual knockout, you can somehow avoid that. There are different ways to do virtual knockout. It's called stochastic silencing. Another idea is to using different framework. We use autoencoders to have a graph neural network in there, and have a different pipelines achieve the similar goal, but use a different framework. So this is a nucleic acid researcher published. Okay, so next two. Okay, so let's take a time. So next one is about cell-cell communications. I will very quickly go through how these communications can be inferred. And this tool is called XCT, stands for interaction. So say you have two cell types in the same sample, from the same animal or human sample. This is a fibroblast, and this is a dendritic cell. How they talk to each other. So this fibroblast will create a small peptide or ligand produced and excreted to the between cell space. And then this dendritic cell will have a corresponding receptor to receive that information, the concentration, et cetera, et cetera. That's through the ligand receptor mediated communication to crosstalk between cells. That's the way. So what we are going to do at a single cell level is to look at the expression level for this. For example, we are talking about CCL19 to CCR7, this pair of ligand receptor. We will see whether fibroblasts express this ligand in a high level. Correspondingly, the corresponding receptor CCR7 will also be upregulated or highly expressed in the dendritic cell. If it is highly expressed, make a pair, then we can see, okay, this fibroblast cell type A have a high level of ligand, and its corresponding receptor is highly expressed in cell type B. In dendritic cell have a level 7. So the product of this 21 give you the how strong this communication is supposed to be. It's not real, but it gives you some hint that this crosstalk can be enhanced by the larger number of this product of this concentration. Okay, so if you believe this is from a ligand will give from a cell type 1 and give 2, if you do this way, it's opposite direction. So if you have cell type A gives the ligand and the cell type B gives the receptor, then you have a different way. Anyway, so you can use this product between ligand and receptor expression cross two different cell types to infer the communications. But we don't consider this case because the same cell type, same receptor is expressed in the in the different cell types, only receptor, there's no ligand, then we don't consider this pair is a good combination. So this is like an example. In the real case, we consider this sample, the stellar cell and the endothelium cell, the purple cells and the yellow cells are cross-talking to each other, and we can have ligand receptor pairs to look at this. So this is the pipeline we use to build detection of this ligand receptor communications level. See which pair of, there are many pairs, different pairs. So which pair is dominant from this particular sample. So this is one sample, but we classify the cells into two different sample types. You have a type A and a type B. From type A, we will find ligand receptor average, and then we will find the corresponding receptor expression, so to make corresponding matrix out of it. But first, we will build a cell type specific gene regulatory network. By far, you already know how to build a network, and build two of them. And in the middle, there are ligand receptor communication, this corresponding matrix to indicate each pairs of ligand receptors expression correspondence, and give you the so-called interaction level. So put them together. So this one network with a corresponding matrix. So network can be expressed as a matrix, adjacency matrix, and the three matrix will be concatenated, make a block matrix symmetric, and use a neural network to learn and to project them into the manifold. In here, we are going to look at the distance between ligand genes in one cell type against its corresponding genes expression, the projection point in another cell type. If they are close to each other, small enough, they are considered to be cross-talking. The communication strength will be considered as a higher compared to others. So if you have thousands of these ligand receptor pairs, this procedure will help you to get those top ones. Those are the stronger candidates to undergone these communications in the cell samples. We have cell type A and B. So those ligand receptor pairs will be prioritized. And also you can search in the database, or some of these ligand receptor pairs have been validated experimentally before. There are real biology talk between them. You can also discover those pairs which not in the database. So this is examples crosstalk between ligand receptors. So this ligand, this receptor between fibroblast dendritic cells. You can build a network, and what's inside this each cell type themselves, what other genes are regulate or which other genes are underregulated with these communication partners. We can also have a multiple cell type get involved. You can do the pairwise detection between any pairs of this. Our paper has been highlighted with for a review paper, which spent a lot of words on describing us. So what we are doing next is try to simulate if you block these communications. We are not knocking out any genes. We just simply block this cross cell type communications pathway and see what will happen. Because we know there are a lot of communications, for example, between, in this case, will be tumor associated tumor infiltrated macrophages with the T cell. If you block some of the crosstalk, and the cell toxicity effects may be stronger to kill the cancer. So we try to simulate for our next tool will be CKO, this is communication knockout. This is ongoing. My students are working on that. So those are the examples we did under this network construction framework, the tensor manifold suite, and the network comparison, virtual knockout, communication detection, and simulation of these drug effects to block the communication between cell types. So those are the students, and Yongjian from ECEN, and Daniel also. Okay, so now we are switch. Again, I still have a couple minutes and to completely change this to a quantum world. So we believe what we can move next is to replace the whole framework and each to use quantum computing framework to do this. So we look at the quantum network already published. We are working on this one here, communications. There's some very good progress recently. And this is relatively easy if we have this. Okay. Salim is my postdoc. He's working in this particular field. And this we call it quantum systems biology or quantum single cell systems biology. If you call the quotation around it, you Google this field, there's no individual site will have that hit. So very, very new. I consider it as a future direction combined with agent AI recently. So it's a very cool area. So let's first very quickly jump to the AI field we're working on. We try to build a self-driving hypothesis generation AI system to help us rely on the RAG and MCP most recently skills in order to construct this AI system to do this analysis I just described automatically based on existing data sets. So this is a RAG system, and you know this retrieval augmented generation. So basically you use language model to ask questions. The language model will retrieve the vector database which contain the embedded information from their training information. The information we will get is from a geodatabase. This is a NCBI repository for single cell, for a lot of gene expression analysis data. So each of these, this file is extracted from a single cell experiments with a description of the experiment setup, cell type, and all the information have been retrieved. And we use these combinations of this to guarantee all the studies as single cell studies. And in the same time, we can automatically download this data and process them and automatically. So all the data has been already pre-processed. Annotation will be done automatically, depends on the human data or mouse. And we go through every individual downloadable and processable record from a single cell study. And this is one example. We have neuron fibroblasts, the different annotation, and those are different. So you will have thousands of this pre-existing data sets ready to be used. So we put meta information and process the data and make a framework. This is called GeoCeller, and contains over 80,000 of the studies, high quality, because anything we cannot process considered to be low process, low quality. And there's more than 500,000 of the samples, either from human or mouse. The information metadata being vectorized, put into Chrome database, and the relationship also there's a SQL database as well. So you can do the search, you can start to ask questions. Then the agent, their hypothesis generation agent, and also the experiment design agents to set up the pipelines, and we'll collect those data together and to do this, what I just described, 24-net, KNK, XCT, or DEDV analysis, all the pipelines automatically. And hopefully they can generate new hypotheses and come back for the for loop to get new information and synthesize to build new results. So this is one example. In this through the dialogue, I ask the green ones myself and ask, study lactation and the breast cancer relationship. Then the downstream will be the GPT. This is the cloud. Start to give. Once you give the instruction, this particular agent can start to search the database and find the relationship, and then goes into the database and found the sample studies which can be used to test the hypothesis, their samples IDs. Then it starts to say, oh, let me try found studies first with the breast cancer, then anything with the lactation, then tumor. Then this is a study founded. They are unrelated studies. So the authors of these two studies, they are separated, maybe one published before another. But because of the context of this hypothesis, maybe they can drag together to compare. So and then I ask, please give me a testable hypothesis. Then the hypothesis will be built. Then data analysis plan will be proposed. And so we need 4 groups from this sample and the process QC and then normalize data, and then to do this Borker's gene expression analysis. Anyway, so this is just to give you a sample of how this part will be able to achieve some automatic hypothesis test. So quantum computing is very new stuff, and nowadays, because people are building the machines, and there's a lot of applications in the chemical, financial field, material, and optimization field, and utilizing superposition and entanglement to do things. That's a Nobel Prize recently. So classical computer can do this, but if you have a hyperspace and all the states can be described simultaneously, and the inference will happen, then you get the results. Hopefully, one of these will stand out. One of the states will encode the information for the solution. So this is an overall framework, how you utilize the algorithm. So that's a paper we published in the intersection of the single-cell biology and the quantum computing. So this is a quantum gene regulatory network, and this is in the quantum machine learning journal. It's about a quantum annealing approach for the cell feature selection. And this is most recently, we have simulators to simulate the data, be ready for the detection of the cell-cell communications. Same idea. So this is a build gene regulatory network, and this is using Boolean network to do things. This is in quantum computer, you simulate genes with qubits, and then try to build, for example, there's multiple genes, then you can have a cell state network. And this means all the genes are shut down, and this means the first gene expressed, second gene not expressed, third gene expressed, something like this. And then you look at how many cells are in this states, how many in this, and so there are many states where there's no samples, no cells in that particular state, but you can somehow infer the change of the state's frequency and map back to the relationship between genes, how you achieve that particular attractor. That's the idea for the paper, and that's the expression for the core, the kernel of those regulation. We try to optimize parameter in the entanglements in order to achieve the similar distribution of the spectrum of the cell states in order to reconstruct the regulation. That's the examples. How many minutes I still have? 2 minutes? Yeah. Still have 15 minutes. Good. Okay. So this is an illustration how you use individual qubits to simulate gene expression. So if this block sphere representation of this qubit is switched on in the 0 position, so this, when you measure it, it will give you rates of 0 for 100%. So this is like a fixed outcome. Well, if you have states for this qubit to be pointed to the one position, then you measure it, and you will have a one condition, the states 100%. So this give you the simulation. For example, this gene is always on in the cell. Then you have this using this state to represent its condition. You can also change the states of the qubit into this superposition through this hard-mount gate. Then when you measure the qubit from the superposition, then you will get 50% of zeros, 50% of ones. So I will use a qubit looks like this to simulate a gene which 0.5% expressed in a cell and 0.5% switch down. So if you collect the 100 cells in the sample, you will have this gene on in 50 cells. In another 50 cells, this gene expression should be 0. So I use this qubit to simulate the frequency of activation of the gene in the population of cells. So you measure it, you have to get this frequency. You can also use a rotation, so this is called RY gate, to rotate a certain angle in order to achieve, say, 75% of 0 and 25% of 1. So you can rotate this and give a certain angle to simulate a gene which 75% of silence, but 25% will express in the cell. So you can control this angle in order to achieve all different kinds of combinations of activation level. So this is only one qubit, but if you have two qubits simulated together, if you put both of them into superposition and then to measure the joint states, and then you will have 4 equal likely states. You have 1, 1, 25%, 1, 0, 25%, 0, 1, 25%, 0, 0, 25%. So this, you can use this 2 qubits to simulate a population of cell. You only consider 2 genes, and you found some cell have both genes expressed. So you have 1, 1 cell in this bin. You may have a cell which only express one of the gene, then you put that into this bin. If your population have 2 genes which completely unrelated, and they all have a likelihood to express 50%, that's a situation that you're supposed to observe in the population, the frequency spectrum, and also you can use this 2 qubits to simulate these 2 genes. But how, if you can link them, this is called a controlled RY gate to introduce the condition. So when this gene, this qubit have a state equal to 1, then you will, through this gate, this will change this particular qubit to a certain angle to change that expression activity for the downstream. So if you build this control gate, integrate into these 2 independent qubits, you introduce the correlation. So if you introduce a correlation, you will have a change in the spectrum. You can change those parameters to how strongly linked to fit your data. If your data have this 0, 0, 0, 1, 1, 0, 1, 1, slightly different frequency, and you can change the parameter here to match the observed frequency spectrum. So that way, the information of this angle will give you the knowledge of this, how 2 genes are associated in this particular simulation environment. So why we need that? We found this the level of this expressivity, the ability to express this high-level interactions or hyperdimensional space much easier. And if you want to change one gene and have an impact on all other qubits, and if you have a fully connected, fully entangled system, this Hamiltonian expression will be much easier to simulate. And if you, this is a very powerful way to simulate data. So it's good to build a generative model using quantum system to generate all kinds of scenario situation to have the, if you have a Bayesian framework that will give you a landscape, all the possibilities to do things in contrast to give you a strong signal out of this noise background. We can do this in MATLAB. People use Qiskit and Python-based a lot, but we realize MATLAB have very strong quantum computing support package. Even you can do Shor's algorithm using this MATLAB environment. So the Shor's algorithm is only algorithm in the whole field that really work and show the super primacy of the quantum computer. All the other algorithms are either suboptimal and or just the junk. So there's only one algorithm in the field, which. So we try to build a killing algorithm from a single cell perspective. Maybe utilizing our method can get slightly better results compared to not using us, but this may be achieved to a new treatment or more precise medicine. So we are using Hamiltonian to stimulate the gene regulatory network to find a dynamic response. This is a perturbation, and this is our next goal, and try to see whether their network change shift from one state to another can help explain the cell state change transition. They all undergone this simulation. So this is a cell-cell communication, so we can simulate the genes, 3 genes from cell type 1, and let them to communicate with 3 qubits in considered to be cell type 2. So if you build a linker between them, this crosstalk link between 2 cell types. Those are the qubits, 6 qubits to simulate this. And we do have a data set which we can use to validate whether our simulation makes sense or not, or we can detect whether there's a communications between cell types. The data we use is from a cancer-associated fibroblast with, I believe this is a tumor cell, a cancer cell. And they crosstalk through those, we read the paper and summarize what a candidate ligand receptor pairs we should use and come up with this model. And this model contains 3 genes from a fibroblast side cancer cell, contains 4 genes. There are internal links between them. The crosstalk is between these 2. And we can simulate, because they have data from a monocultured and co-cultured system. So they put, in biology, they put a cancer cell isolated and culture them. You have a expression like this, and you have a monocultured, so isolated fibroblast culture. Then they put them together and then see, okay, what will change. So that's a crosstalk can be validated experimentally. So we build this and build the profile of those genes against those genes and see whether with interaction or without, what will change in terms of those observed patterns. So we can infer the crosstalk between cell types. Okay, so MO stands for monoculture and CO stands for co-culture system. I still have time. 5 minutes? Okay, so very quickly, and those are the gate-based. So IBM has, Google has, now we have this D-Wave, have a quantum annealer. So they are specialized to do the annealing computation. I know D-Wave also have a data-based quantum computing, and they just purchased another company and try to do data-based quantum computer as well. But this study, we use a D-Wave system to use a quantum annealing to simulate the system to in order to, so they provide this solution, or you can have an interface and access to their machine. So this is to do the regression for single cell data for feature selection. So in this case, this is single cell gene expression matrix, big X, and their rows are the cells and columns are the genes. Well, we want to have a feature. So this feature is what we know. For example, we know the evolutionary time for the cell. So this is from early stage and later stage. So we want to find gene combinations, say which gene correlated with this particular feature. Okay, particular target variable. So genes are the features. We want to select from thousands of genes, have a particular combination, maybe 5, maybe 15 genes. When they combine, explain this continuous variable the best. So this is a kind of regression. So we build this R matrix using neutral information. Then they have this vector. So you combine this and you compute a Q matrix. So this Q matrix is in the center of the Qubit framework. And once you have this, you give this to annealer. Annealer will give you a feature vector and one indicate that the gene will be very useful. And the second 0 means the second gene not very useful. And the third gene very useful. So there are combinations of features and to help you to select the cell, select the genes. So the system we use is from embryo stem cell and have a very clear development trajectory to differentiate into the endothelium cell. So you follow this trajectory to indicate, to assign values for the starting point as 0 and then end as 1. Then it continuously you assign all the cells in this trajectory different time. And we want to find which gene responds to push the cell from the stem cell to endothelium cell. If you use lasso, you will find those genes expression is almost always monotonically increase or decrease with the time. Well, you have also this called the random forest regression. You can see that some. But the Qubit give you a very unique solution. It's combinations of many linears and with very some of these nonlinear combinations. So each of them is illustrate a slightly different categories or group of the regulators. So we found, for example, there's some monotonically increase, decrease, but you also found this S shape go first. So those genes at the end we found is very important to drive this differentiation. So this is an example to use quantum annealer to perform feature selection in order to find the most responsible gene to drive the differentiation in our case. Okay, so this is my lab, and over the years, not so many people at this moment, and there's some funding agent and also connections with the data science institute here. Thank you for your time.
Learn about imaging?
Yeah.
So tissue level.
Tissue level, yes.
Can we really integrate single cell information with tissue level? I mean, you touched base a little bit. With
spatial, yeah. Yes, spatial transcriptome. Yeah, so it's the technique is still evolving right now. So last year we have a resolution which not strong enough to get enough spots, but then this year there are high resolution of spatial transcriptome. We do have enough data to be processed in order to understand what's going on there. Yeah. It's another, yeah, absolutely, yes. It's ongoing efforts on the campus we have to help people, yeah. Yeah, thank you for saying that.
All right.
Okay.
Again,
Thank you.